I'll quickly give a brief introduction about myself. So I'm Prashant, founder director of IncepBio. So IncepBio is a decade old year company now. So we are operational more than 10 years now with 120 employees across three countries, that is India, Singapore, and Malaysia. Predominantly focusing on three service lines. One is validation calibration, which is for qualifying all the equipments, clean rooms, even the smoke studies across all the pharmaceutical and hospital segment. And then comes our C&Q, that is commissioning and qualification validation, running the projects starting from the initial stage till the facility delivery. Again, this we are doing across all the three countries. And finally the turnkey, we take up the complete projects starting from your designing, facility erection, installation of all the clean room panels, AHUs, then also qualifying all the things and closely working with DCDS, CDSU, and also USFDA to get your facilities and processes qualified. So this is about our services, and that is about me. Over to you, Gaurav Dikshini.
Yeah. Thank you, Prashant. And I think all the three service line that what you are talking about is very, very important at this point of time, because many new facilities are coming up. Many of the existing facilities are having challenges in FDA and other regulators. So I think that's the great services that will help them to grow there. And of course, the ultimate bottom line is to serve the patients worldwide.
Yeah. Yeah, true. Agree with you. Agree with you.
Yeah.
And coming to the today's topic, Gauraji. Uh, so, what do you think? In general, because you are an expert in airflow visualization, and I'm sure you have trained so many industry professionals regarding this. So, what is your top level thought when it comes to challenges in airflow visualization? Just a general thought. Then
Yeah.
one more good thing I want to update you. Like we have got so many questions from even our audiences today. So we have made prioritize those questions. So I will be asking those questions to you, and to all our viewers. You can also type your questions in the chat box so that we pick those questions. We'll be also asking Gaurav about those things. Yeah, Gaurav, over to you.
Yeah, thank you so much. So I will start introducing myself. So basically, by qualification, I'm an engineer and the instrument and control, MBA in business analytics. And then I started my career with Cadila Pharma, moved to Dr. Reddy's, Chemwell. Lastly, I was with Cipla. And today, of course, airflow visualization topic, which is very, very important, because many of the people says that, okay, I do all the HVAC validation, right? I do ECPH, I do air velocity, I do particle count, I do the recovery test. Then why do I need to do the airflow visualization? That's a major thing. And the people have the misconception that when I do a lot of things on validation, even static and dynamic, everything I do, uh, even the temperature, RH mapping, differential pressures, all of those things. But still, then why do I need to do the airflow visualization? So I always talk about the things what you do is like static and dynamic is to show you the state of art about the HVAC, but how the airflow along with the people movement, how the first air is getting disturbed, what is the neutral buoyancy, how the airflow movement is happening. That's when this airflow visualization plays into the picture. So this is about, you can say, the art and science of HVAC, while the airflow visualization is something which is intersection between the operator, the movement, the process, and HVAC all together, put together, how the things will go. That's when the major challenges come into the picture.
I'll just start with the first question. What is the single most critical principle in airflow visualization?
So the first thing, whenever I see the airflow visualization or any auditor sees the visualization, they want to see the first air. So let me just show you in the presentation, so it will be good for the audience as well. So let me just share the screen. Basically, in the airflow visualization, we always talk about why, how, and what. So which is a golden circle of airflow visualization. What exactly is a airflow visualization? Why it is to be performed? How it is to be performed? And when it is to be performed, that is very, very important. And coming to the question where the first air. So the principle of first air, like if you think from the perspective where the air is coming from the HEPA filter, it should touch nothing else but directly the open containers or the open product, that's when the first air is coming to the picture. And that's the most important principles, because we must document the evidence that when the product is coming, the airflow is not touching anything else. It is flushing away all the particles, and it is now touching nothing else but the open product containers. So it is touching nothing else before it flows over the container. That's the most important principle of let's say the airflow visualization, because you can have the very clear let's say video where it is showing the flow is coming, it is going somewhere, it is how it is behaving, everything is good. But unless and until you prove that your air which is coming from the out of the HEPA filter touching to the containers and nothing else in between, that's the most important part that you can able to show it to the auditor, so which is very very critical for the manufacturing of the sterile products.
Yes, it definitely makes sense, Gauravji, that first air is something. So that is what people should first focus on. That is the most important principle of airflow visualization studies. Okay,
Yeah,
thank you. I'll move on to the next question.
yeah,
What type of smoke is scientifically acceptable for airflow visualization?
yeah. So typically, when I started my career, at that time we used to use the water fogger. We used to
Exactly.
use the glycol based for foggers. But
Yeah.
now, typically in last couple of years, what I have seen, like last 2 years, 5 years, the US FDA auditors or majority of the people are talking about neutral buoyancy.
Okay.
Now, if I explain, it will be I mean, any picture shows the thousand words. So let me just show you that particular slide, that how the neutral buoyancy, the difference will come. So now this particular has got two videos. Like there are two
Yeah.
videos which are going to be parallel. In the first video, you can see that clean room is off, like the air is not there, and
Mm.
here the clean room is on. Now,
Okay.
if there is a neutral buoyancy means there is a zero gravitational force which will come onto the particles, and it will not allow the particles to follow the gravity path. Now,
Okay.
when you see this video, and then after I will explain you more about this particular part. This particular video, even though left side there is a clean room, air is off. It's a completely there is no airflow which is pushing the tracer particle to go let's say bottom. Still, the particles are flowing on its own, and that's when the concept of zero buoyancy has come into the picture, because water foggers or some of the oil-based foggers, their particle thickness, even though they, which is let's say zero point 0.1 micron to 1 micron, which is required, even though they are very heavy in the weight, and because of that, even though there is no air, it will still follow the gravitational force, and it will go let's say from top to bottom. And this is where it is like a false positive. Like, of course, you are a
Okay.
microbiologist, you know
Yeah.
what's a false positive result. So this is something which is going to happen when your particles weight is itself is very high. So it will, because of the gravity itself, it will go down rather than it will follow a natural airflow path. And
Okay.
so zero buoyancy is something which now are very popular. Like there are, courtesy to Microrite and the other players in India, those who are doing this particular part, they are following that particular, let's say, a proprietary material which will have the particles with the zero buoyancy, which will follow the air tracer. I mean, the tracer particle will move, let's say, move with the air rather than just following the gravitational force. Yeah, so that's when the major difference comes. And because, of course, we need to find out, it should faithfully follow the airflow streams, not
Yeah.
just because of the false positive result it should create. Yeah,
Understood. So what you were suggesting is, so any chemical which is having this zero buoyancy and it is not
yeah,
giving you false positive results. So that is something, what you should be adapting to your entire airflow visualization studies.
Yeah,
Okay.
yeah, yeah.
Okay. So, is it like only one particular brand, like Microrate you were mentioning, or are there any other alternatives
No,
also?
there are other sources as well. So that is something which has to you need to check at your end when you are switching off the AHU or switching off the clean room air, and then
Hmm.
see the particular movement of the air. It is not only one particular brand like which I mentioned, but there are certain other brands as well available where we are working together with them as well, and that can also give the similar results. Yeah.
Okay. Great. Great. Thank you. So, I think, this is something very important. Okay. So something which is having zero buoyancy, and we understanding in a age to switched off condition, and understanding whether the gravity is having the impact on these particles
Yeah.
or it is able to maintain. Okay. Understood, Gaurav. Thank you. So the next question is also similar related to that.
Yeah.
What is a buoyancy test and why it is important?
Yeah.
And is
So,
it mandatory? That is my question.
yeah. uh, Nowadays, looking at the 483, so I'll answer the last part first, and then
Okay.
what is buoyancy? Because of course now by this time we understood now how the buoyancy test we can do. Uh, you say whether it is mandatory or not mandatory. I would say it is not mandatory. It is recommended as long as we are able to prove that the tracer particles are moving with the air rather than its own by gravitational. So definitely, if you are able to prove that, then it's great. But it is not
Okay.
mandatory. It is nothing. It has not mentioned in any of the till now. It has not been mentioned in any of the guidances or any kind of
Okay.
regulatory. But of course, it's like a GMP. It's a CGMP, like current good manufacturing practices. So people follow a good manufacturing practice, and based on that, people are saying that okay, it should have the zero buoyancy in the chemicals. I mean the chemical what we use, and the tracer particles should faithfully follow the stream.
Okay. Okay.
Yeah.
Great. Okay. But have you also come across any of the remediation work, USFDA remediation work or warning letters in which was also predominantly on airflow visualization?
Yes. Yes. Absolutely. So there are many 483s which I have seen recently. Uh, like they majorly it starts with three things. One is your lighting. Second
Mm-hmm.
is the aseptic processing, which is a very, let's say, like airflow visualization. If you divide that equation into three parts, the
Mm-hmm.
80% of the part is your aseptic processing.
Correct.
Then 10% is about your lighting and the camera angle. So 10% each, I can say. But majority of the part is your how you're putting your cameras, like how you are doing the aseptic processing and visualizing that camera part. So majorly, I would say around 70-80% should go into your aseptic processing. Around 10-20% should go into your camera angle, your lighting, and all of those things. So majorly, I have seen like operator is leaning over the particular place into the, let's say, OREPs and doing the, let's say, intervention. And during that time, the first air is getting cut or first air is getting disturbed.
Mm-hmm.
There are certain 483s that I have seen is people are doing the open door intervention, and during the open door intervention, grade B air is moving inside the grade A,
Oh, okay.
which is also one of the another uh, that particular part. Yeah.
Okay. So is it a problem with the conventional RAB setup or
Yeah.
something
It
like that,
is,
right? Okay.
yeah, it is majorly the design issue which I have seen.
Mm-hmm.
Like whenever there is a pre-filter just on the top of, let's say, your OREPs, so the air is getting short-circuited. The air which is moving bottom also is getting short-circuited. So majority of the time, what I have seen remediation, like you are asking about the remediation, I have seen people are now moving from, let's say, LF base to the FFU, which is a fan
Okay.
filter unit, and
Yeah.
there the air which is sucking from the AHU return rather than the room return
Okay.
from the pre-filter which is just on the top of the LF. Yeah.
Okay. Okay. Great. Great. Thank you, Gaurav. So I'll move on to the next
Yeah.
question.
Yeah.
What are typical design issues uncovered by smoke studies?
Let us see this video and then answer the question. So now the question to all the audience, and I want all of you to type in the chat, what do you see in this video? What was wrong thing, and what should have been the correct thing in this video? So I can see a couple of chat as well. Is the airflow velocity verification required simultaneously during airflow visualization? Okay, answer done by Ashish. Thank you, Ashish. Is it required to do buoyancy study for LF units? Is it enough to do in the clean room? No, separate buoyancy study is not explicitly okay. Okay, so I think yeah, great. The questions are getting answered simultaneously. That will be great. Now, if you see here, let me just take a pen as well. Yeah, if you see, there is a complete dead space here. There is no air flowing, and of course, your entire work is happening in this particular space. And if you see, the air is moving towards this side. Yeah.
True.
So now you see, this is where my critical area is. But if you see, this is a complete dead spot. Yeah. So that's when, like, you have done the HEPA filter integrity, HEPA filter velocity, particle count, all of those things are okay. But where you are working, whatever this workbench is utilized for, maybe a washing, maybe whatever, I don't know exactly about this part, but this is something which is, let's say, a critical area, and there is no airflow which is moving in this particular direction. So that's when the airflow visualization will give you the clear picture that if there are any dead spots are there, is there any issues with regard to equipment blocking. Let's say one of the time what I have seen, like in one of the workshop which we have done together at your site at IncepBio, I have shown one of the smoke study where there was a table, there was a wall, partition wall, and then there is a table which is touched to the particular wall. Air is coming on the top of that table and then getting swirled.
Oh.
So the air is coming and it is touching on that. So
Okay.
in the like in the facilities, we always say that don't block the return air, don't touch any table directly to the wall. Otherwise, there will be a reverse swirl will come. So we should have some space so that air will pass through it rather than bouncing back again into that part. So rightly asked by you, a lot of things like equipment having blocking the first air. There is a misalignment in the FFU, or are there any glow ports which are shadowing the isolator, and a lot of engineering design failures that I have seen when you do the smoke studies, and that will only come when you do the smoke study. It will not come with the regular HVAC validation.
Thank you, Guruji. Very nice video to explain this particular problem. I'm sure like this, it will be showing lot many other, labs of a entire clean room or
Yeah,
equipment. So by
yeah,
doing the airflow visualization study. Okay, great. Thank you. And the next question is very important. Something what even I used to observe a lot when I was working in the industry, that we always see any particular study or process from an auditor perspective.
yeah,
See, our main aim should be, okay, look, this is something we have, either the process or the engineering system or the quality systems, which we'll write in a way which is followable, and then we execute in a way which can be done and documented. Okay, so always we
mm,
see from that perspective. That is ideal, wherein your entire system is solid, and that is something what you usually even FDA looks at.
yeah,
Whatever you have written, are you following or not? But also, most of the companies, the management will drive the people in a way, look, do everything as USFDA needs. So many
yeah,
of the times we keep overdoing the stuff, rather than there are hardly two, three things we should be doing, but we end up doing 8 to 10 things. And that is where again FDA will catch us. Look, why you have you done this? And if you have written this, why this was not done? So people go in that direction. So the next question is the one which I am sure many of the people will be very much interested in. What do FDA investigators specifically look for in airflow visualization videos?
yeah. So as I always say, and this is like a statement, challenge your aseptic processes, not just to showcase that, okay, I'm great in doing my airflow visualization. So always challenge yourself that, okay, whatever I'm doing is something which is written somewhere. Is it something to be asked by someone, or it is something which it is going to challenge my own process then you do it, not just to please the USFD auditor or please any kind of management. So that's when whenever we interact, like I had a good opportunity when I was with Cipla to interact with a lot of USFD inspector, and even of course when I was at Zydus.
Okay.
What I have seen, typically people, the USFD inspector, they just want to see that whatever product is going to the US, they should be safe, and the it are patient consuming, they should be able to there is a whatever that CSPQ, like that particular 5 elements of the qualification, they are met. So what they see in the airflow visualization is typically whether my first air, which is coming, let's say from the HEPA, it is touching nothing else, but it is directly touching to the particular containers throughout my process. Even I'm doing intervention, even I'm doing, let's say, any kind of door opening, door closing, or if it is that's isolator, or if it is that's ORFs, close ORFs, whatever things are there, they just see the first air is coming and touching nothing else but that containers. That is the first thing that's true. Then another thing what they see is the air which is coming from the top and going to the, let's say, containers, and then where it is going. Is it bouncing
Okay.
back? Is it coming inside into the OREPs again, or is it getting flush through the outside? Where it is going? So starts from the origin of the air, big to the, let's say, reverse, whatever the return air is there. That entire path they follow. Another
Okay.
thing, what they see, whether the simulation has happened in the worst case condition. If it is not done in the worst case condition, then might be whatever they are seeing, that is not a worst case. So if it is a, let's say, biggest container that you are using for visualization, you are, let's say, using the largest container, or the, let's say, biggest container with the more time, like, because it's a residence time which is going to more contaminate during the entire process. So whether the time which is used for you to simulate the entire process is what you are actually using for the worst case scenario. So that is another thing. And then, as I told you, that in one of the things, that when operator is opening the open door and doing the some kind of intervention, the grade B air is going inside. So those kind of cross contamination between the two grades, like grade B and grade A, are there any visible things? As I mentioned about the complete smoke path which is going,
Yeah.
and then of course they will also see your entire aseptic processing, like
Okay.
how whatever is written is that what you are following. One typical example I want to give, uh, like one of the time in one of my old company, I don't want to mention the name, but let's say the auditor was Peter, and he wants to see
Okay.
that I want to see the complete disinfection of the entire filling line that you do. He has not told anything why he wants to see.
Okay.
He has done the entire disinfection,
Hmm.
and then he see that how much time it took by the operator when I am seeing,
Okay. Okay.
and how much time typically you are taking.
Understood. The actual
So
scene, and then the
yeah,
auditor is seeing it. Okay.
yeah. And then what he has done, he has compared both the data.
Both the data. Okay.
And none less to say, but of course, when our auditor is seeing, it will take more time than usual. So
No.
you
Also,
know.
you do more mistakes. Operator tend
So
to do more mistakes.
yeah.
Okay.
And
Okay.
then typically what happened is he came up with the argument saying that, okay, when I'm seeing it taking this much time, when you are seeing this much less time,
Hmm.
so what is the correlations? And of course, it is very difficult to explain
True.
all of those things. So that's when it happens. So like they see a lot of things when they are of course there for visualization. So during that time, how much time it took, how much time in real it is taking, how much time when
Okay.
I'm doing, and how the aseptic processing and all of those things.
Great. This follow-up question to the previous question. So,
Yeah.
why do regulators often question the camera angles and the lighting used in smoke studies?
Yeah, because like light refraction is a major principle which I want all of you to understand. And let me just pull up the slide for the camera angle and lighting, because that's a major challenge people are facing, that particular part. So if you see in this particular part, the difference between the reflection and refraction, because
Okay.
whenever the light is passing through any medium, like whenever we are putting the light angles, we are putting all the lighting and everything, the lighting will transfer through that the smoke medium, and that's when the difference will come. Some of the times, even though you are doing correctly, even though the smoke is following the properly, but when the lighting angle and the camera angle, that gives the different picture. So what is a reflection? It is bouncing back of the light from a surface. So if there is a, let's say, mirror, and on that particular light is getting reflected, then you are able to see. But it will not
Correct.
happen the same thing when if it is passing through different medium. Like there is a smoke, and the lighting is passing through that, and at that time the reflection is going to happen, and you will not be able to see. Like this is one of the example where if the light is passing through a glass and the water, two different medium, you will be able to see the straw at a angle or bended. You will be able to see that it is bent something.
Yeah.
And that's when the principle comes, when you are putting the lighting, camera angle, and all of those things. And that's when the auditors wants to see that is there any masking is happening because of the, let's say, camera angle, or if there any, let's say, poor lighting is not showing me something which is I need to see. There are certain wrong angles, or if there is a too dense smoke, like one of the 483 which I have seen,
Hmm.
the smoke is very dense. Smoke was
Okay.
very dense, and because of that, what happened was you are not able to see.
So here,
Yeah,
this my personal question. Like, there's a lot of human factor in the way you are suggesting how it should be seen. But
yeah.
is there again there is a particular lux or something which we should keep in mind uh, when the light is coming towards the camera? Is there something which we need to? Usually we measure the lux of the clean room lights, everything. So
Yeah.
is it something like that, or is it entirely on the experience?
I would say there is no any fixed data where you need this much lux. Like in normal condition, clean room require, let's say, 400 to 500 lux for proper working. That's for the general work. But for, let's say, a kind of airflow visualization, there is no kind of any fix that, okay, you need minimum this much lux. Only thing is it should not be able to let's say, blur the entire operation what is happening. Like if you see this particular video, you can't see anything.
Okay.
There is a lot of fog inside this, and you can't see anything. So I mean, there is no kind of fixed limit that, okay, you should have this much lux. But of
Yeah.
course, it should be able to, you should be able to showcase something which you want to show to the
Okay.
auditors. Yeah.
Understood. Understood, Gaurav. Thank you.
Yeah.
Let me ask the next question. So, how do smoke studies link to contamination control strategy? CCS under Annex 1,
Yeah.
especially all the with the new Annex 1 implementation, everything, and CCS becoming a big thing. So, how
Yeah.
exactly this is correlated to that?
Yeah. So basically, CCS come into the picture in 2023, in
Yeah.
somewhere in August 2023, and then the draft came in 2022 August, and then of course 1 year it was given for review and everything, and then it has come into the force in 2023 August, if I'm not wrong. And what it typically talks about is that you should, like till now we used to do all the risk assessment. Like we do the risk assessment for the process, for the people, for the method, and all the risk assessment. But when the CCS came into the picture, it talks about a next version of risk assessment, that how your facility is controlling the contamination through various, let's say, controls. So
Okay.
airflow visualization is one of the great tool which I have seen, because any risk you see, until and unless you visualize that risk through the airflow visualization, you can't be able to control. Like the example which I have shown for the dead spot, the example
Yeah.
which I shown, let's say, where the first air is getting disturbed by the operator, the outside air is going inside. Until unless you see this visually through the airflow visualization, you can't say that, okay, my contamination control strategy is better, because on paper it looks better with regards to
Yeah.
HVAC validation, with regards to all the other things control that you have. But when it comes to practically seeing the airflow visualization, you will be able to showcase more that is there any dead zones are there, is there any contamination transport routes are there which is transferring the risk to the, let's say, the product, or is there any operations which are happening. Like I have seen, typically people are now moving from open door intervention to the closed door intervention, and that's why the isolators are becoming more popular. They are moving from OREPs to the isolator. They are moving even if there is no possibility to, let's say, move to isolator for the existing lines, they are moving to the closed door intervention rather than open door intervention, and that is all happening because of airflow visualization with the CCS. They are both going to help justify the entire the part. And regulator expect that APS finding to be referenced in CCS as a risk assessment and justify the EM locations and aseptic controls. That's what the major requirement which will come from the CCS part.
Okay. Can high air changes per hour, okay, compensate for poor airflow patterns?
Yeah, so this is one of the question that if I do airflow visualization, I mean, why should I do airflow visualization? Because I
True.
already have ACPH, a very good ACPH, let's say all of those things. But if it's see, ACPH and all of those things are a macro level metrics where you will be able to see your ACPH is more than 100 for grade B, uh, 30, 60, and all of those things for grade C and D and all. Uh,
Mhm.
Micro environment behavior, which will come only by the smoke studies, because your smoke study will reveal any micro environment issues which is going to come up because of the lot of intersections between operator, process, let's say OREP movement, HVAC, so a lot of those things. So that's when I mean, macro level versus micro level. That's when the difference comes. It is not going to compensate even if it is a high ACPH. It is just increasing the turbulence, but not
Mhm.
giving the right protection.
Okay. Okay. So when we say this, is it only for critical equipments, that is fill finish area, or do you also, it is recommended that even for other critical clean rooms, though it might not be fill finish area.
Mhm.
So is it recommended to do the smoke study for both the type of areas, or will you do it only for the critical areas?
No, smoke studies are always required for the critical areas, where the critical areas you can define from where your, let's say, contamination can going to come into your product. So let's say if
Okay.
I start from grade D, grade C, grade B, and then grade A, some of the time people are having, let's say, isolator facility, then they will have
Mhm.
not grade B, they will have the grade C and then grade D. So
Okay.
depending upon the facility design, you need to
Hmm.
do your own contamination control strategy from where the contamination is going to come. But of course, let's say airflow visualization, which I have not, which I typically believe that you should be able to showcase that whatever the processes you are following from, let's say, grade D till your final product which is coming out, you're
Hmm.
in a state of control with regards to the contamination, and that will, like, facility to facility, it will change process to process. If, let's say, someone can argue that, okay, I'm having terminal sterilized facility, I am
Hmm.
not having aseptic, I am having terminal
True.
sterilized. So
Yeah.
it
Why
depends.
do you care even for airflow visualization, right? Because it's
Yeah,
a
yeah.
TS product. Yeah.
Yeah, TS product. So it depends, like how much your, let's say, contamination control strategy, risk assessment, what kind of, let's say, different aspects that you want to work upon that, and then you came up with your own strategy in a proper documented with the justification, which will be, let's say, buy in by the let's say US FDA regulators. Yeah,
Okay. Okay. Uh, Next one is very important one. So people do this both static versus and dynamic, both
yeah,
airflow pattern visualization studies.
yeah.
Does it matter even today that you have to do both? Is it mandatory, or why don't I do only the dynamic one?
Yeah. So this is very, very good question, Prashantji, because people have the misconception that, okay, I need to do only dynamic or any, let's say, only static. What should be the typical difference? So typically, US FDA, the FDA requires, let's say, in operation to be done only. FDA doesn't talk about that. I want to see the static. But while
Okay.
EU is something, EU is something which will recommend that you should do in static as well and in dynamic as well. So if you are, let's say, a facility which is let's say, focusing on FDA regulation, and you are not going to anything else in that particular facility, then you should only do, let's say, in operation. But while
Okay.
the EU is something which is recommending as per the guideline that it should do static as well as dynamic. So that's when the requirement will be fulfilled. So static means you are not running the product online. Majorly you are used to qualify the HVAC, while dynamic is something which is an actual simulation of your product, where you run the actual run, of course, with the W5 water, not with the product. And then you can identify what are the things to be, let's say, is there any dead spot there? Is there any eddy currents? Is there any, let's say, reverse flow? All of those things you can able to control. And of course, with the number of persons performing the activities should be the same, because dynamic is something where you should define that, okay, I will have minimum 6 people, or let's say maximum 6 people in my area. So you should be performing that particular part. And it should be like a worst case. The protocol should define clearly what is the worst case scenario for each of the line.
Okay.
Container size, speed of the machine, number of operators, number of intervention should be the maximum. So all of those things, yeah, will be required. So my personal recommendation, static and dynamic, both we can do, because static will give you something which is in principally all the things are correct before I'm doing any kind of operations without running the line. Dynamic will give you the entire picture. But if you ask me about the guideline, US only talks about only dynamic. Even if you're not doing static, they will not give you any 480 on that. But if you are going for EU, definitely you should do both.
So in principle, so what you're suggesting is the static gives you a fair amount of idea about the room and the equipment, the performance
Yeah.
in general.
Yeah. Yeah.
And the dynamic is something you should exactly mimic the situation which is used during the routine process or production operations.
Yeah.
And also consider all the worst case scenarios.
Yeah.
Correct.
Yeah.
So
Absolutely.
both are always required. Okay, understood.
Yeah. Yeah.
The next point is like what happens in a product is getting released, so there is always something assay. So it means assay of 90%, 95% above, it passes, it fails. Microbiology study passes, it gets released, it passes. BAT passes. Now, how do you say that a airflow pattern visualization study is a pass or a
Yeah.
fail?
Yeah.
How do you say this?
Definitely. So this is a very good question in terms of understanding what is pass and what is fail. The Let's say if I give you a very short answer, there is no 100%, let's say, a correct answer by anyone whether this particular study is pass or fail. Like today, I mean, in one of my earlier companies I've seen, one audit happened. One auditor came. He saw that particular smoke studies, like few videos out of them. He said, everything is perfect." Within 6 months, there was another audit which was triggered by another regulators, and then it has given, let's say, multiple points in that. So
Okay.
there is nothing like a complete right or wrong, but there are certain few checklists that we have designed,
Okay.
and like that checklist is something which will cover all the points when you are, let's say, reviewing the smoke studies. Because before you say it is a pass or fail, you need to review the entire smoke study. So review part will have the checklist. The review part should have the cross-functional team like engineering, quality, validation, let's say quality assurance, quality control, manufacturing. All the people should be a part of that, and they should be able to review the entire smoke study, if not possible together, but at least at an individual level. And at that point of time, they should be able to see particular things which is written in the checklist. Like if I say, which is, let's say, pass, like in case of HVAC, you can say my ACPH is a calculation, it is like a quantity. Yeah. uh, NVPC is pass or fail based on, let's say, if it is less than 2900 particles, or zero point 0.5, less than 20 particles of 0.5, let's say 5 micron, then it is a grade A. You can claim that as a grade A.
Yeah.
That is like a quantity, and velocity is something which HVAC qualification prove. In case
Yeah.
of smoke studies, it is the air behavior at a product level.
Yeah.
So sterility assurance depends upon the first aid protection, And
Hmm.
not only the HVAC data, but your entire functional performance airflow visualization, that will be able to tell that whether this is a pass or fail. And you
Okay.
demonstrate the unidirectional flow, you demonstrate that critical zone is under the, let's say, first air. There is no, let's say, reflux, turbulence, ADs, dead spot. Is there any compromise on the first air? There are, let's say, needles, bowl covers, and all of those things are, let's say, clearly you can able to showcase that the things are going from the top till the, let's say, the return of the return HEPA, return air. So that is something which we can say as a pass.
There is one quick question, however, though I saw in the chat box. That's a very good question. The person was asking, should this be done periodically? Like, I can put this in many perspectives. One of them is whenever there is a major machine part change, okay, or whenever there is a process change. Of course, we also check all these things during media fields, but also if there is any huge engineering activity which has been happening in the place. So, how often we should be doing this?
Yeah, so this is typically as per the guideline, it should be done, as you rightly mentioned, if there are any major changes which is happening. Apart from that, periodically it should be happening. And I have typically seen around 1 year to 2 years. In some of the companies, they follow 2 years strategy. In some of the companies, they follow 1 year. But typically, airflow visualization for the critical area, you should be doing periodically, of course, because uh, even though you say that nothing has changed in last 1 year, 2 year, you should be able to see because your blowers are aging, your HEPA filters are getting age, your pre-filters are getting age, your operators are changing, your let's say return risers are getting blocked,
Yeah,
your movement of portable instruments or portable tables are moving. A lot of things are changing, right? Even though you
yeah,
say that I have not changed many things, but you can't say that it is not changed because definitely there are things that are going to change.
yeah.
As I told you about all of those things, so you should be typically doing periodically depending upon your risk assessment.
Okay, okay. This one of the last questions from my end.
Yeah.
What is your one-line advice to avoid 483 observations related to smoke studies?
Yeah. So basically, as I earlier also told that design your smoke studies to challenge your own aseptic processing, not
Mhm.
just to showcase or please the auditors. Okay, I can do a very good video and I can do a multi-angle videos. Wherever I have seen, like one of the things, like one of the person asked me, do we need
Mm.
all the videos to be multi-angle? Like there
Yeah.
should be a multiple cameras only. I said that if one camera is sufficing your need, then why do you need multiple cameras?
Also, there are many other people who sell this idea. No, you have to do this. Also, it is like very expensive, so it is very good. It will avoid 483 observations per view. This webinar is also for the same reason. Like we should focus on the logic, why this should be done, how it should be done, and when and all it should be done. I think now we can move to the questions from the
Yeah,
audience who have put in the chat box.
yeah. So what is the requirement to do in the clean room at an operation condition as it doesn't have a complete LF? We have only HEPA. It doesn't cover the entire area. How? If I have to answer this question, I would answer that what is the requirement to do in the clean room at operation condition? It is a guideline requirement. USFDA says that you need to perform the airflow visualization in the dynamic. EU says that you need to do in static and dynamic. So that's mandatory when the it's regulation. It's not a guideline, it's a regulation. USFDA says means it has to be done. Uh, as it doesn't have complete LF, even if it is not a complete LF, whatever the things which are covered under LF, you need to perform that part. We have only HEPA, it doesn't cover the entire area. So I'm not sure about the facility, whether it is aseptic, terminally sterilized, or what kind of facility you're talking about here. But if I assume if it is a filling facility with the grade B, with grade A environment, grade A with backed by grade B, you have to perform this particular study, and you need to showcase that first air is touching nothing else than the product containers, and airflow which is coming from the top, it is where it is going and there is no eddy, there is no reverse flow, all of those things.
There are two more questions. What I had, like, if we
Much.
perform AFP in line with EU requirements, do we need to perform a static condition study only during filter replacement, after modification, or after new installation, or it is required periodically as well? So this is asked by Mr. Uday.
Yeah. So as per the EU, you need to do the static and dynamic both. Uh,
Mhm.
but static typically you can do for the first time when you are starting the facility, or let's say if you are doing a major change, like you mentioned about the filter replacement. I don't think so. Filter replacement is a major change.
Okay.
Typically after that, because static is only done during the initial stage,
Mhm.
not let's say after filter replacement. You can do only dynamic, uh,
Okay.
then post, let's say, initial installations.
Okay, great, great. That is what even something I was thinking in line with.
Yeah.
And the other question is the area containment test to be performed by using smoke study or any different method? I have a query. Can I ask after this question? Yes, Prasad, can you please unmute and ask the question quickly?
Yeah. Yeah. As we told, we need to simulate worst case interventions and everything in the smoke study. So is it required to simulate number of times door opened and closed? Like for example, I'm doing some intervention in the grade A area. Like other some operators are there, they open the door and close to bring some metals and out. So is it required to simulate like this interventions also, other than my routine interventions? Like these are some kind of event.
So in the intervention, if you go through the guideline, they talk about two kinds of intervention. One is a inherent intervention, which is by default, which is going to happen, and something another type is like a exigency, or something which is like a let's say out of the requirement, or something like which is you can say extrinsic, or something which is something has happened and because of that you are doing. So
Yeah,
when
yeah,
it
some
talks
breakdown.
about a, yeah. So in inherent operations or inherent things like rubber stopper is stuck, seal is stuck, or let's say your vial has stuck somewhere or vial has broken. These are like something like which is a inherent things there you should do. There you should have to keep a check on it, that okay, whatever the number of operations that you do as a part of, let's say, media field study, which is like you during your routine operation, you will be having, let's say, 5 or 10 of these during a particular period. You should be able to simulate that during that one. And even something which is a very extrinsic, which is like a maintenance of, let's say, a gear uh, screw or something there inside, the maintenance of something which is not a routine. So routine intervention, non-routine intervention, both you should simulate during the let's say airflow visualization, to come up with saying that okay, I have proved that with these interventions, I'm not, let's say, yeah, having the issues with my first air. I'm not having issues with my, let's say, any eddy current or reverse flow or any kind of thing. I should be able to prove that part. So
Okay, okay,
routine, the correct word is routine and non-routine interventions. You should be able
okay.
to simulate both during that part. Yeah.
Yeah, thank you. Other than that, recently we have faced FD audit. Their auditor is requesting for the raw data. So he
Okay.
required spoke study raw data. So
Mhm.
I didn't see any guideline which is saying to store raw data. Yes, we do have smoke study videos which capture details, but still he want raw data, how it is captured. So uh, what is your
So
view on
raw
this?
data,
We need
when
to
when you
maintain
say
that
yeah, so when you say raw data, what
uncut
they were
videos,
asking,
uncut
on
videos.
editor, on uncut videos. Okay, yeah.
Hmm.
So it depends, because I when I see when I have faced a couple of UFS FDA, it was not being asked that I needed that particular raw data, but it depends auditor to auditor. It depends how the things are presented, or if they want to see something in detail about any particular video, then definitely, yeah, they might ask.
Okay, okay, thank you.
Yeah.
And Gauravji, so this we always talk about, aseptic processes is very, critical for visual inspection, like visual airflow pattern study. How about the similar studies when it comes to a drug substance or a API processing unit. So they also have the labs, they also have dispensing units surrounded by grade C, grade D areas. So what do you think? So this is also one of the audience question from Mahadio Katimanni. So the question is it the same criticality matters even for DS?
No, I don't think so. So when you say drug substance facility, if it is you are claiming that as a sterile or because there are sterile APIs, there are non-sterile APIs. So what do let's say take that particular part? If it is let's say sterile API, which is not going to process at a let's say at a pharma company and directly it is getting, then definitely the seriousness will be different or let's say the kind of controls will be different. But if it is, let's say, non-sterile API, non-sterile facility, just a LF is to protect as a grade D and grade C, then I don't think so. You need to do in that criticality of the process. But of course, static is something which I recommended to do, even if it is a DS facility, because that will tell you a lot of things as a design. And of course, dynamic, it depends upon the criticality and risk assessment, and of course, the final product, what we are claiming at.
Okay.
Yeah.
Okay. Great. And there's one question from Anandra. What do current regulations such as EU GMP Annex 1 and US FDA state regarding door opening simulation during airflow visualization? Should the airflow be demonstrated with the door fully open or partially open, like 75%, 50%, 25% door opening? Since different clients follow different as a validation service provider, how can we standardize our approach when client expectations differ from one side to another?
Okay, very good question, Anand, right?
Yeah.
Yeah. So I'm sure you are talking about this particular door is between two different grades or within two similar grades, not the OREPS doors, right? This particular part is as a part of just to show that the air is flowing from the higher grade air, higher grade to the lower grade. That is what something, if I can see it properly. But I have not seen typically people are saying that 100%. See, when it opens 100%, the differential pressure becomes zero. Between two different grades or even with the two different same grades, as per the guideline, if it is a two different grade, you need to maintain 12.5 Pascal differential pressure. If it is, let's say, two similar grades, you need to bear minimum 5 Pascal, you need to maintain. That is what the guideline says, and you need to follow that part. So when you open the door, let's say 25%, 30%, or 50%, you'll be able to see there is a movement. But when you say open 100%, when the door is fully open, typically it will balance out at one point because the differential pressure becomes zero because the volume is such a great, you can't see a flow which is going. I have typically seen people are opening maybe 5%, 10%, or max up to 25%, and they show the let's say the airflow which is going from the higher grade to the lower grade. But one more important part which I want to share, one of the incident from the previous audit, where the camera angle is so close onto the door, you can't see whether you are standing outside the door, whether the flow is going towards the other side. You are not able to see that. So the auditor comment was that you are so worried about just a smoke, you are not showing me the full picture. So I always tell my people whenever I give the training, as we do a lot of training for airflow vigilance, I tell the people that you first start with the broader picture, where you are standing, in which side of the smoke is there, and then you go to the zoom. Rather than doing other way, like you first zoom that and then unzoom, you first show the entire picture. What is the door, let's say nameplate, what is the room number, all of those things you show properly. Then you start the flow and then you zoom it just to showcase how the flow is moving. Yeah, that is more important. Yeah.
Okay. Okay, Gaurav.
Yeah.
Thank you. The logo, the skill up to scale up, is
Yeah.
something what we strongly believe at IncepBio too, that we always need to skill up our skills every year. We need to elevate ourselves to reach out
Yeah.
to different destiny and manage bigger projects, bigger clients. Please do suggest us, send us your feedback. wherein we can upgrade ourselves, change ourselves the way we do this. Okay. And thank you all for joining this wonderful session. Thank you, Gauravji, for delivering such an amazing session. Thank you all.
Yeah,
Take care.
thank you all. Thank you all. Thank you, Prashant ji.
Yeah.
And let's see you in the next one. Yeah.